Hello and welcome back everybody. Today we are going to solve the paper 2 multiple choice October-November 2025. Okay, paper 2 variant 1. Let us start. First question is telling you diagram shows a plant at the start of an experiment and the same plant 2 weeks later. Start of the experiment, as you can see, if I made the light from one side, as you can see here, after 2 weeks the plant already will go towards the direction of the light. So basically I can see that the plant had increased in length. So basically this is growth and sensitivity, because as you can see, as the light was moving to that direction, the plant moved to the opposite direction and it moved towards the direction of the light. Okay, so basically I'm not here explaining about an oxygen fact or something. It's only a basic question. Of course it is being growth and sensitivity. Number 2, what can be used to classify organism? The number of sequences of bases in the DNA is the correct answer. Number of bases in the DNA is actually nothing to compare or to classify organism, because basically they will have the same bases. They have adenine, thymine, cytosine, guanine. There is nothing like an extra base for you to say it is correct. So A is incorrect. Number of different types of bases, the same thing, it is already adenine, thymine, cytosine, guanine. Nothing is different. Okay, sequences of bases in one protein. Well, we are not classifying upon the protein, we are classifying upon the DNA. So basically 2C is the answer. Here is telling you diagram show a single cell from an organism called this name. Which feature does this organism share with plant cells? So let us see now. As you can see, it shares a cell wall, it shares a vacuole, it shares nucleus, it shares cytoplasm, it shares chloroplast, it shares it shares everything. So we have 1, 2, 3, 4 and 5. So basically the answer is A. Nothing from this table will be unsatisfied. Everything is satisfied and every answer is well done. Now how to get this? We have the formula I am, and you can use this triangle, basic triangle, and just calculate your calculation. Okay, but remember that this calculation, for me to occur to be like this, M is equal to I divided by A. Okay, so where is the image length? The image length is 5 centimeter and the actual length is 5 micrometer. So basically if you make it like that, you will get the answer 1. But in order for us to get the same answer, we need to convert that centimeter to micrometer for it to apply. So how I'm going to do that? First of all, I'm going to multiply by 10 and to convert the centimeter to millimeter. So basically let us make it here, centimeter to millimeter, and then I'm gonna multiply by 1000 to convert that millimeter to micrometer. So basically the answer will be 10,000. And how I did it, basically let me explain it for you here. So he's telling you that we are going to follow the triangular method formula. So I am. So in this question, in this case, the actual length we were finding, what we were finding, let me check it. We were finding here the magnification. So in order for us to find the magnification, we will rearrange the formula. So it will be M is equal to the I, which is image length, divided by A. So it will be 5 centimeter, as I saw in the question, 5 centimeter, and you need to multiply by 10,000. Why 10,000? I'll tell you later. Now divided by 5, which is what? Which is the actual length. Okay, so basically I'm going to multiply by 10 because centimeter I need to convert to millimeter, so I'm gonna multiply by 10. And for millimeter to be converted into micrometers, I need to multiply by 1000, so a total of 10,000. So we're gonna multiply that 5 centimeter with what? With 10,000. Okay, so this is just an explanation here for you to memorize it and understand it as well. So let me just edit it for you. Yeah, this one like this, and it like that, like that, like that, and like that. Okay, now moving on to the next question. Question number 5. Well, number 5 would be tricky, look to be tricky, but it actually is nothing. Okay, so let's just read it together. The potato was used as a source of the plant tissue. For potato, sticks of identical size were cut from the same potato. At the start of the investigation, each potato stick was 10 squares long. Please focus with this. 10 squares long. This is the original size. 10 squares long. The potato sticks were put into 4 separate petri dishes. Each petri dishes contain a different concentration of sugar solution. So apparently, first of all, I need to consider my brain that the potato disc must be 10 squares long. I cannot deal with any question unless it should be a 10 squares long potato. And then it's saying you each petri dish contain a different concentration of sugar solution. Now you will ask me, well, does the question require you to know the concentration? No at all. Let's see. They're saying you here which dish contains a sugar solution, okay, that has a higher water potential than the potato tissue. It has a higher water potential than the potato tissue, meaning that the potato tissue will increase in length because basically water will move by osmosis down water potential gradient and it will enter into into the potato. So basically I need that 10 squares to be more than 10 squares, and let us count them one by one. So starting with D, which is the right answer, I can see here 1. I can see here wait a minute. Yeah, I can see here 1 square, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 11, or 10 and a half. So I can see that they had increased in length. So the potato tissue had increased in length. So I can see here 1, I can see here 2, 3, 4, 5, 6, 7, and 8, and 9, and 10, and a 10 and a half, let's say 11, okay? Now for that condition, which is C, I can see here, I can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 9.5, let's say it's 10, for example. Now it's telling here 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. So this is complete 10. So basically I can see here that this one is 10.5. Wait a minute, let me count first the letter B option. 1, 2, 3, 4, 5, 6, 7, 8. So now let us make it a conclusion for you to understand. As you can see here, that one has a total of 10, that one has a total of 8, that one has a total of 9.5, and that one had a 10.5. So basically the one that had increased in length was letter D. Now question number 6, which row shows correct statement about active transport? Well, active transport, as you know, it requires transport against a concentration gradient, because down a concentration gradient is diffusion, and it is not the case in our question. Requires a partially permeable membrane? Yes, of course, because let me define active transport. It is a transport of ions or mineral ions against a concentration gradient, okay, through a partially permeable membrane, okay. So you need a partially permeable membrane for active transport to function. So now moving on to the next question, which is question number 7. It's telling you here the graph show effective temperature of an enzyme controlled reaction, and this is repeated a lot now. It is repeated a lot in May, June 2025. Which statement describes the effects of temperature on this reaction? So in this reaction is telling you here, as the temperature increases to 25, which I can see the 25 in this part, okay, here the reaction speeds up. So 21, 22, 23, 24, and now it's not like this, each line is by 2, so 22, 24, 25 is somewhere in between here, 22, 24, and 25 is somewhere in between here. Yup, like this. So this is 25. So this reaction is speeding up. That fact is true. So A is really right. Question letter B. Between 10 degrees Celsius and 20 degrees Celsius, the enzyme denatures? Of course no, because actually the rate of reaction is still increasing. The rate of reaction is not decreasing, okay? So as the rate of reaction is increasing, of course, between 10 degrees Celsius and 20 degrees Celsius, the enzyme won't denature. No, because after the optimum temperature is exceeded, that time the enzyme will denature. But before that, of course, it will not be denatured. Now, between 35 and 45 degrees Celsius, frequency of effective collisions increases. Well, let me check it. Now 42, 44 and 45, 42, 44 and 45 is in between here. As you can see that the rate of reaction had decreased, meaning that the optimum temperature, which was in this case 30 degrees Celsius, had been exceeded. So basically the rate of reaction would decrease. Now question number 8. The graph shows the rate of photosynthesis of a plant in full sunlight in different concentrations of carbon dioxide. All factors were kept constant. What limits the rate of photosynthesis at 0.02 carbon dioxide concentration? Well, we're gonna look at here. So the question is telling you to look at here. So basically, if you look at here, like that, here the graph did not yet level off. So the x-axis will be what? The x-axis will be your carbon dioxide limiting factor. So carbon dioxide concentration will be your limiting factor because we did not yet reach plateau, or we did not leveled off. So carbon dioxide concentration would be your limiting factor. But in a case it here asked like starting from 0.06 to 0.14, what is the limiting factor? Of course it will not be carbon dioxide concentration, it will be something different. Question number 9. A student wrote some statements about palisade mesophyll and spongy mesophyll tissues. Which statements are correct about description of two tissues? Okay, let us check it. Spongy mesophyll tissue is near the stomata for gas exchange. Yup, that's true, because as if you can see, like, let me draw it for you here. The drawing for the epidermis and everything. So I have here my epidermis. I have here by palisade mesophyll cells. Palisade mesophyll cells, they are rectangular. And then I have what? I have my spongy mesophyll cells with many air spaces. Okay, so as many air spaces. Let us for example, that yellow is the air moving in through the stomata. So the spongy mesophyll cells. This is just like an illustration for you to understand. Now here it's telling you the palisade cells are closely packed, but there are air spaces in the spongy mesophyll cell tissue. Okay, as you can see, I have like illustrated for you like an accurate. See here, as you can see, the palisade mesophyll cells, they are closely packed, but in the spongy mesophyll cells they have air spaces, which show that statement 3 is right. You will tell me why statement 2 is incorrect, because statement 2 is telling you here only palisade mesophyll have chloroplasts for photosynthesis. Well, they don't, not only palisade mesophyll cells, because the guard cells, for your information, guard cells have chloroplasts to carry out photosynthesis. So they are not only palisade mesophyll cells, also spongy mesophyll cells have chloroplasts. So it's not like the only palisade mesophyll cells have chloroplasts, okay? The fact that makes students confused in this question, because they think that palisade mesophyll cells like the only one having chloroplast. No, the correct information is that the palisade mesophyll cells, it is the most part that have chloroplast, but it is not the only part having chloroplast. What is the role of a bile? Okay, so bile here, it emulsify fats increasing the surface area. Okay, so of course not, it does not contain enzymes that digest starch, because enzyme that digest starch is maltase, and is not the case here, or amylase, I'm sorry. It kills bacteria in the food? No, it does not kill bacteria in the food. It provides optimum pH for pepsin activity? No, because stomach provides optimum pH for pepsin activity, not the bile, which is secreted by the liver. Okay, now, where in the alimentary canal is the enzyme trypsin found, and what are the products of the reaction trypsin catalysis? Okay, so enzyme trypsin will be found in the duodenum, where the pH is pH 8. Okay, the pH for that trypsin working, it will be pH what? It will be pH 8. And products, it will be amino acid, because basically trypsin catalyzes amino acid breakdown. It hydrolyzes protein breakdown into amino acid, which are the basic subunits of a protein. Question 12 is telling you here what is the structural feature of xylem vessel? Okay, it's made up of cells joined end to end. First of all, like, let me tell you that A will be incorrect, because xylem vessel, it is a dead vessel. It is what? It is a dead vessel. And I'm sure that in paper 4 you had learned that xylem vessels are dead vessels, so basically they won't have any mitochondria, and they won't have cross walls. So what are cross walls? Well, cross walls are this. So I'm having here a cross wall, and that is a cross wall. That is what is a cross wall. Well, I don't have cross wall, they have free, like as you can see here. Wait a minute, let me just make it for you clearly. Yep, they have this part freely, so water and like can move freely here. Water can move freely and easily. Thin walls, of course, no, they have thick walls to withstand the high pressure of water flowing through them. So 12 will be answer is B. Question 13 is telling you which, during the grass growing season, tomato plant produces flowers and fruits. Which parts of a growing tomato plant are sources and which part are sinks? So it's a growing season. Let's guess it is a spring season. So basically the leaves will be the source, and the leaves will provide the fruits and the flowers with the amino acid starch that had been produced during photosynthesis, the glucose that had been synthesized during photosynthesis. So all of that will be provided by the leaves, because the leaves is the one who's carrying out the photosynthesis. So basically 13 will be A. 14. Which statements are correct descriptions of method of preventing pathogens from entering the body? It's telling you here platelets clot blood. Of course, the one which is fibrin is converted to fibrinogen to form a mesh. That when there is a wound, and that is not the case here. Okay, phagocyte produces antibodies. Okay, but like this information also is not relating to the question. It's telling you preventing pathogen from entering the body. I want to prevent it from entering the body. So let, for example, see if I having here like a skin, okay, and it has been torn. So what the platelets will do, it will provide like a covering for it to prevent the pathogens. To prevent what? To prevent the pathogens from entering. So platelets clot the blood. It is the only like option which is truly correct. Okay, and that one actually one is true, is like it is relating to platelets, but it's not asking you for the way how it is being formed. It's telling you what is being formed, not how. Okay, so basically 3 will be correct, and 2 and 4 are things which occurs inside the body, not preventing your like pathogens from entering into the body. Now here 15. Which statement describe as double circulatory system? Double circulatory system meaning that the blood goes through the heart twice every time it goes around the body. You will tell me why not lungs? Because basically it does not go through the lungs like twice. It only, as you can see, if I having here the lungs and having here, so the deoxygenated blood will flow out and the oxygenated blood will flow from the other side. So I don't have it twice. Through the kidneys twice, of course, no, not through the kidneys twice. It is the same as the lungs. Only oxygenated blood pass and deoxygenated blood come from the other side. It does not move through the heart twice. But in the heart, of course, it flows twice, because as you can see in our heart, as you can see in the heart, we have a deoxygenated blood. So I'm having here my 4 chambers, okay? So deoxygenated blood is entering in here. Now it will flow through the lungs, okay? It will flow through the lungs, which will be in here, and then the oxygenated blood will flow again into the heart to be pumped out by the left ventricles through the aorta to all parts of the body. So as you can see, there is a double circulation. 16. The 3 statements about immunity are listed. Specific antibodies are produced in response to specific antigens. Okay, pathogens have specific antigens. Of course, each pathogen have a different and its own antigens, which is like linked to it. A vaccination stimulates an immune response. Of course, a vaccination stimulates immune response against viruses, because bacteria are being killed by antibiotics, and I had explained that in the May-June 2025 variant 1 question paper. Okay, 17. Which muscles in the breathing system contract during inspiration? Inspiration, so these things you need to memorize. External intercostal muscles and diaphragm only. These are memorizable things, nothing to explain. And here, which process require energy from respiration? It is protein synthesis. Protein synthesis requires energy from respiration. Okay, of course diffusion is not, because diffusion is passive transport. Osmosis is passive transport. Detranspiration, it is the evaporation of water, so you don't need energy to evaporate water. Evaporation of water can occur due to high temperature present in the surrounding. That's it. Question 19. What is produced by anaerobic respiration in human muscles? Produced by anaerobic respiration in human muscles, will be lactic acid only, because as you can see that aerobic respiration produces carbon dioxide as it is waste product, but anaerobic respiration produces lactic acid, and water is not produced in anaerobic respiration because it is a thing which does not involve oxygen. Question 20. Which statements are correct for a healthy person? blood leaving glomerulus has the same concentration of glucose? Of course no, of course no, because as the blood entered it will be 100% absorbed in the proximal convoluted tubule, so you cannot say that the blood leaving glomerulus will be the same as the blood entering glomerulus. Of course no, because as the blood enters containing glucose in the proximal convoluted tubule, as it passes the BCT, which is proximal convoluted tubule, all of the glucose will be 100% absorbed. It's telling you in 2, there is no glucose in the fluid leaving a nephron, which is the explanation to the 1, so it is the same. A nephron absorbs all of the water and some of the ions filtered out of the blood by a glomerulus. No, it does not absorb the water by the glomerulus, it absorbs the water by the collecting duct and loop of Henle, not by the glomerulus. So basically 2 will only be the right choice. It's telling you here, when moving from an area with low light intensity to an area with high light intensity, how do the circular and radial muscles of the iris react? So basically the radial muscles will relax and circular muscles will contract, because we need to narrow that opening of an eye. So basically if I have here my eye and I have here my circular muscles, and I have here my radial muscles. So basically the radial muscles, which are these lines, will, if it contracts, okay, it will pull that iris, will pull that pupil eye out there, it will pull the pupil to be larger. But if the radial muscles contracted and the circular muscles, like, they will narrow around it, narrow, it will cause that pupil to be narrower. So causing the pupil to be narrower will cause it to have the eye closing to prevent damage from the high light intensity. 2022, the graph show a change in the percentage of infections caused by antibiotic resistant bacteria in one country. Telling you which statement could explain the change in the percentage of infections caused by antibiotic resistant bacteria. So the answer here is letter B. Why? Let me tell you. The use of antibiotic has resulted in natural selection for resistant bacteria. Of course, anything that you are going to use for a longer period of time will cause it to become resistant. Like for example, when you are taking Panadol, okay? So the excessive intake of Panadol, so the more you take Panadol, your body will know, the bacteria knows that you are taking that Panadol, so it will develop resistance against it by natural selection, which takes over like many generations and take a lot of time. Now here, the use of antibiotics when not essential. The use of antibiotic when not essential increase the percentage of antibiotic resistant infection, okay? So it's telling you here, the use of antibiotic when not essential increase the percentage of antibiotic resistant infection, because basically the more the intake of the drugs you're taking, the more the resistance will increase, the more the antibiotic resistance will increase. So basically 1 and 3 are correct and number 2 is incorrect, because it's telling you the use of antibiotic has caused a decrease in viral diseases, which I mean that are caused by viruses. And when I'm talking about viruses, I'm talking here about vaccination programs. I'm not considering anything about the antibiotics. I'm not considering anything about the antibiotics. I'm considering only the thing which is talking about the viruses. So a viral disease will be basically it will be solved and resolved by a vaccination program. Antibiotic only kills the bacteria, okay? So this is a point we need to keep in our mind. Question 23 is telling you here, the gardener has a strawberry plant that produces large sweet fruits, okay? How does the gardener grow more strawberry plant that all produce large sweet fruits? So the gardener, of course, he wants to increase his crops, to increase the yield, to increase the price of his yield. So basically, for us to answer such a question, of course we'll use asexual reproduction, because you cannot use sexual reproduction, you don't have gametes. You don't have gametes. So you will use asexual reproduction to produce offspring with no genetic variation. You don't want any genetic variation, because any genetic variation will cause these characteristics to change, and you don't want the large sweet fruits to be small and non-sweet fruits. You don't want that variation, you want it with nothing to be changed. So basically A will be the correct answer. It's telling you here where does fertilization occurs in a plant. So fertilization basically occurs in a plant in an ovule. Of course not a pollen grain, because a pollen grain will be carrying your pollen grain will be entering into the anther, so this is not our case. Pollen tube, of course no, pollen tube is the place where the pollen grain will move through it. A stigma, no, because stigma, it is the It is just like for receiving your pollen grains. It is nothing like more. So ovule, where this is fertilization occurring in a plant, other parts are really nonsense for them to be an answer. Now here is telling you which organ secretes most hormone X in the late stage of pregnancy. So basically, organ which secretes it, it will be the placenta. Okay, it will be the placenta because, as you know, that the placenta releases progesterone, okay, with aligning with estrogen. So hormone X, of course, it will be placenta. It cannot be the vagina because vagina does not release any hormone. Uterus does not release any hormone. Ovaries only releases estrogen. Estrogen will be released in the first of the stages. As you can see here, that the estrogen hormone, it is released in the first case, in the first stages of the reproduction. So it is not our case in right in here. Now a person has a 28-day menstrual cycle, in which day is the lining of the uterus to be thickest. So lining of the uterus to be thickest will be in this case only, as you can see here. In 28 days, in 28 days or 21 days. So as you can see here, if I draw annotated in here, so here is 28. Let's just say that this is 21. So yeah, so basically from the sources that he had provided you, 21 will be the most day where the uterus of lining will be thickest, because see, 2, it will be menstruation. Number 4, it will be like reactivating or like rebuilding your uterus wall. C, which is 9, it also will be rebuilding, so it is not the case in here. Now 27, which statement describe the sex chromosome in human gametes? It's telling you here X cell contain only X chromosome, okay, and this is actually true. And now, because as you can see now that the female has only X and X. Remember that the female has what? Has X, and the male, that is a female, the male have X and Y. So just beware for this information, because it's really like beneficial for you to understand and memorize it, because it comes in degree questions, it comes in many questions. Question number 3, sperm cells contain only Y chromosomes? Yes, because I just draw here for you the diagram showing that the female have only XX chromosome, but the male have only the YX chromosome, okay? So basically 2 and 3 will be right. Why 1 is incorrect? Because X cell only contain X chromosome, it cannot contain a Y chromosome. Number 4, here is telling you here, sperm cells contain only Y chromosome. It will be incorrect, of course, because sperm cell contain X and Y, so 4 will be incorrect. So 3 is telling you sperm cell contain either X or Y chromosome, which is actually the right option and correct option, because sperm cells contain both. 28 is telling you here which statement describe the effect of meiosis on chromosome number. Okay, meiosis means what? A diploid parent cell reduces haploid daughter cell. So if I'm having here 46 chromosomes, they will be split into 23 chromosomes. Basically, like meiosis is dividing by 2. The chromosome number you had provided me, okay, meiosis will be divided by 2. If it is mitosis, okay, it will be the same. So divided by 1, and it will be the same. Nothing will be changed, nothing will be reduced in the chromosome number. So basically the answer will be B. Why it is not C? Because it is not haploid. First, I'm starting in meiosis, I'm starting from a diploid cell, like a parental egg, okay, an egg from the female and a sperm cell from the male. So both of them will have what? A diploid number of chromosome. During meiosis, I will cause these sperm cells to become having a haploid number of chromosomes. So when a haploid sperm cell and when a haploid X cell will mate and fertilize together, they will produce a diploid zygote. Okay, that is it about the question 28. Now 29, a female with red-green color blindness has a child with a male who does not have that case. The child is male. What is the chance of this child having red-green color blindness? So what is the chance that this child, which is the male child, having a red-green color blindness? It is 100% chance. Why 100% chance? Because as you can see here, the female has what? Has a red-green color blindness. So let's say now that the female is what? Is X, okay, XA and XA. Now this one will be for a carrier case, but in our case it's not a carrier. So now we'll move it with a chance like this: XA infected, okay, infected will be crossed with what? Will be crossed with a person that does not have what? That does not have the condition. So I'm now having a child that is male, right? So basically the mother will provide me with what? With one chromosome, and the male will provide me with what? With another chromosome. So I have now a male that is infected. So it is infected. So it is 100% infected. Any male will be 100% infected. But if it is a female, of course it will not be infected, because basically I have an XA which is a small and XA which is capital from the father side. So basically the female will be carrier, not infected. Now 30. What is the source of genetic variation that alters the base sequence of DNA? Of course it is gene mutation. Why not mitosis? Because mitosis does not affect anything about the DNA. It just only copies the cell. Remember it like this. Random fertilization, it causes production of a zygote, okay? Random mating, it does not change the base sequence of the DNA. It only causes like genetic variation for the organism to survive, and all thing you know. 31. Which technique is not used to conserve endangered animal species? So of course seed banks, because seed banks are not animals. Seeds are seeds, they are not animals. But A, B, and C are all for animals. Like for example, A and C are used for increasing the population number. B also increasing population number. So of course this question is really basic and common sense. 32 is telling you here which flow chart shows how energy is transformed from the sun to an organism. Now 32 will be D, because energy trapped by chlorophyll will be converted into chemical energy in your carbohydrates, and it will be stored in plants. Okay, it's telling you here why A is incorrect. A is incorrect because chemical energy in carbohydrates will be stored in plants. No, first of all the energy need to be light energy to be photosynthesized, producing your glucose and your chemical energy that will be stored in what? In the plants. 33. Which statements about a food chain are correct? Okay, about what? About a food chain are correct. Now the answer will be here 2 and 3. Let us now understand why. As you can see here in question in letter 1, it's telling you between trophic level 1 and trophic level 2, energy is passed from a herbivore to a carnivore. Well, between trophic level 1 and trophic level 2, energy is passed from a producer to a herbivore, because like the food chain, as you know, starts from a plant. It does not start from a herbivore. It starts from a plant which is a producer, like a grass for example. It starts with what? With a plant. So trophic level 1 will be a plant. So basically you are only left with 2 and 3. So 1 is incorrect, 1 is incorrect. Now he is telling you 2 in question letter D. Between trophic level 2 and trophic level 3, energy is passed from a primary consumer, which is a herbivore, to a secondary consumer, which is a carnivore. Of course, that will be correct. Now, letter D. Now, letter C is telling you 2 and 3. Between trophic level 3 and trophic level 4, energy is passed from a secondary consumer to a tertiary consumer, like for example from a rat, for example, okay, to a tertiary consumer like an eagle or a lion or a cheetah, like for example from an eagle, which is secondary consumer, to a lion, a tertiary consumer. Which process in nitrogen cycle is involved in breakdown of amino acid in living organisms? Wait a minute, it's telling you what? Breakdown. So of course it is deamination. Deamination, it is the breakdown of excess amino acid. You will tell me, but it is in living organism. Of course it is in living organism. So deamination in living organism, like in humans, it occurs in the liver. Deamination of excess amino acid by reducing urea, which will be like transported to be excreted out of your body, right? Remember that. Now still good diagram show about a carbon cycle. Like it's so easy now. Photosynthesis, of course it is D, because the carbon dioxide, as you can see, it is absorbed by the plant for the plant to carry out photosynthesis. So carbon dioxide will be absorbed by the plant to carry out photosynthesis, which is letter D. 36 is telling you here which row identify the phases of population growth. Of course it will be letter in lag phase it will be 1. It is not log. If it is log, it will be 2. Exponential phase, which is also known as the log phase in some books, so the answer will be here 2. Death phase, it will be 4, of course, because 3 phase, it's a stationary phase, and I cannot like say that 3 will be death. 4 will be death, because 3 is stationary and it is actually a graph you need to memorize for your exams. 37. Eutrophication results in a reduction in the dissolved oxygen in streams and lakes, because basically eutrophication is something like that, okay? It's something like what? Like that. The mold will grow here, will grow there, will grow there, will grow there, blocking what? Blocking the sunlight. So basically the sunlight cannot diffuse, the sunlight cannot move into the plants, So basically the plants will not carry out photosynthesis properly. Okay, that's it. Now in here in 37, the answer will be what? The answer will be C. Why? Because increased aerobic respiration by decomposes live inside the river, down deep inside the river, decomposes life. So increased aerobic respiration releasing more carbon dioxide, and that carbon dioxide reduces the oxygen concentration, causing the organisms to die, like fish and other like mammals like plants or other like otters or anything like that. It's telling you here 38. What are reasons for using chemical fertilizers in food production? To reduce the use of herbicides and to increase the crop yield, to reduce competition between crop and weeds. So for using chemical fertilizers, okay, you are doing that to reduce the use of herbicides. You don't just want every day to spray your plant with herbicide. That will be beneficial for people. It will not be beneficial for people. It will be killing your insects, be killing your pests, but it is not the case. Like, chemical fertilizers are not for reducing your use of herbicides. They are not for that purpose. To reduce competition between crops and weeds, no, it's not also for that purpose. Chemical fertilizers are placed to increase the crop yield, to increase the quality of a crop yield, to increase quantity of crop yields, but not at all to reduce the use of herbicides. 39 is telling you here, why is pectinase useful in fruit juice production? Because more juice can be extracted from the fruits. It's telling you cloudier juices be reduced. Actually, that is incorrect because it does not have to do anything with turbidity. Proteins in the fruit are digested, while pectinase is not used to digest the proteins. So basically, to be incorrect, starch is digested to produce more sugar. No, because you need amylase, and pectinase is not amylase. So basically, D will be incorrect. So only B is the correct answer. It's telling you here which stage uses DNA ligase. DNA ligase, it is used to what? To link plasmid with a gene to form a recombinant DNA. So as you can see here, the gene, I have here the gene, okay? That black thing is what is your gene. And it is inserted into that circular thing. And that circular thing is what is your plasmid. So basically, the DNA ligase will join that gene with that plasmid together for you to form a recombinant DNA. But let us check also why others are incorrect. As you can see here in letter A, is telling you that the gene is going to be extracted, and that will be done by a restriction enzyme. In letter B, a plasmid is removed from a bacterium and cut open. This is not used also. This is not restriction enzyme. This is another enzyme which is not DNA ligase. In here, DNA, the plasmid is inserted into bacterium and reproduced. This is amplification. So basically, of course, D will be incorrect, and the only answer here will be letter C. Well, that will be all about the the November 2025 paper 2 variant 1 question paper. I hope you really understood, and if you had any questions, please leave it in the comment section, and thank you for your time, and have a nice day, and looking forward to see you in other videos as well.